does, it's not too bad the way your book approaches it. And so I wanted to highlight kind of the way they're thinking about it and go through some of this with you and engage with it, and then get into the new content. So starting with this metaphorical example, we all know typically how we measure behavior in humans is by looking at the way they behave. And so one way that your book offers, that your Goldstein and colleagues offer, is think about a car. Like, we know that when we push the gas pedal, the car goes, hopefully, right? When we push the brake pedal, the car stops, right? We know those basic things. But we really don't have any idea why that's working unless we look under the hood, right? And so if we look under the hood, we're going to engage with different levels of analysis. Like, we might look at the structure. So how are the cylinders working and the hard pieces that are in there? You know, I wish I could name more car parts, but I can't. So the gearing, you know. And then we could look at more dynamic factors, right? Like the fuel, the coolant, sort of the fluids that are moving through the engine. Maybe that could be something that helps inform us of why the engine works or why it might not be working right. We could look at external factors, like how is this engine relating to things like temperature? And of course, the connections, like the wiring, a loose connection that would cause it. So when we're understanding how a car works, we have many levels of analysis. That's what a mechanic does with his or her knowledge, probably pretty intuitively. So then metaphorically, if we want to understand how our thinking and emotions work, metaphorically, we also need to look under the hood. So we have a subjective experience of thinking, a subjective rising of an emotion, but what's the underneath of it? Why does it work in that way? And so I put an actual guy with a hood here with his brain under his hood. And of course, we have to look into the brain and the central nervous system. And so once again, we have different levels of analysis. Like examining the cylinders in the car, we examine the structure of the brain, right? These two halves of the brain, the two hemispheres. The corpus callosum in the middle there, what's it doing? The different lobes in the brain, the limbic or lower functioning system or primitive system compared to the higher functioning system, the structure. We can look at more dynamic factors, like looking at the fuel in a car. Metaphorically, we also want to look at the messengers in the brain, which are neurotransmitters or chemicals that are produced in the body, right? That are produced through things that we eat and generated through neurons in the brain and are fluid or dynamic, are changing. We might look at external factors, perhaps toxins, or perhaps stressful situations, things that are impacting cognition and thinking and emotion. Just like we look at the temperature when we evaluate the car engine. We might look at connecting factors, right? Like the synapses and neural pathways. And so how are these neurons communicating with each other and talking with each other? And so that's why we have this whole chapter on cognitive neuroscience, because we're looking under the hood right now to see how is the brain related to the mind? What are the mechanics underneath metaphorically pushing the gas pedal or telling yourself to do an action like raise your hand? What's happening underneath to make that stuff happen? So this nerve net was the original idea way back in the 1800s when we first began to understand, at least to be able to look into the brain systematically. Early thinking was that our whole brain network was interconnected, like physically, like a highway, like strings tied together, or like the roads that go all through our country, the interstate map, they're all connected. You know, I mean, the interstates, you could ride on interstates around this country endlessly, and you would always be able to get from one to another. They don't end anywhere. You don't have to take your car over a crazy big bridge or anything like that, you know, no flying leaps. So when they first looked into the brain, that's how they thought things were working. They did Golgi staining, you'll read about that. And that's how they saw these networks, but it was an imprecise method. And so later, when they were able to look at it more closely using other methods, and they discovered the actual individual neurons, they discovered that no, it's not actually like this nerve net. Each neuron is an individual that talks to, ostensibly speaking, communicates with other neurons, of course, through neurotransmitters, but they're not connected. And the whole beauty of that system emerges because that allows our brains to be dynamic and allows us to literally, physically change our own brains. If it was hardwired, if every single network was all connected up, well, that would be it. The pathways that formed when we were young, there would be, that's it. We would be the same throughout our lives. We couldn't learn anything new. But this fact that each neuron is an individual, that's what allows us to learn and grow. So this is just a little review of the structure of the neuron. So this is like the prototypical neuron. They don't all look like this, but whenever you look at a picture of a neuron, you probably remember this well. This is what it looks like. In our next lecture, we'll talk about the interesting fact that every neuron in your brain right now was in your brain before you were born. In very few cases do we experience neurogenesis or the birth of new neurons. Now, a lot of the neurons that were with you when you were born have died, but the ones that stayed with you are learning all the time. And they learn, let me see what I wrote over here. These are cortical neurons, they're in your brain. And similar to that in your nervous system in general, we'll talk about a little bit different ones that take in information from your senses. And we have the cell body. Let me see if my pen's going to write for us here. Let me get my pen. We have the cell body right here. Hang on, let me get my pen again. There we go. So that's this big part of the cell. This is where, say, the DNA is. This is the nucleus. This is where all the information that comes in is accumulated and kind of weighed out to see if there's enough information right here at the end. These little stems or branches are dendrites. And don't they sort of look like the roots of a tree? And what do the roots of a tree do except take in nutrients for the tree? Likewise, these dendrites are filled with receptors. Receptors, receptors, receptors. And they take in chemical information from other neurons. These dendrites, if you're going to learn something new, your neuron and series of neurons related to this are growing. Probably one thing they can do is grow new dendrites. So a neuron with a lot of dendrites is a neuron that is taking in a lot of information and a neuron that you're using a lot. We know that when the signal comes in, it travels down here into this into this part of the neuron where so the chemical energy is transferred into electrical. If there's enough chemical, if it meets a threshold, it's transferred into electrical energy. So our brain, our neurons work using what we call electrochemical energy. So the chemicals come in. If there's enough chemical to meet a threshold, then the neuron does what we call fire, or it experiences an action potential, which we'll look at on the next slide. But right now we're just talking about how the neuron does. And so if this electrical energy is generated, it travels, boom, right through this axon, all the way to the ends of these axon terminals, where more chemical is released to communicate with the next neuron. The chemical is called neurotransmitter, and it's released from vesicles at the axon terminals. Terminal just means the end of the axon. And one thing your book doesn't talk about much, but I want to talk about, is this right here, this white stuff around the axon. This is called myelin. And I didn't write it on this slide, I don't think, so I'm going to just put it up here. Myelin is like the insulation protecting the electrical current. It insulates the axon, so the electrical signal can travel efficiently. And we'll look at a little GIF of this in the next lecture. And you can see these gaps here. There's only one gap in this neuron, but these are called nodes of Ranvier, and the signal sort of jumps over these from one section of myelinated axon to the next. This myelin is really important because if it's damaged, some piece falls out of it, then some of the electrical signal is lost. And so it's possible that there won't be enough neurotransmitter release to continue this message that you're trying to get to your body in some way. So it's a very important protective coating. And when you hear about during adolescence, the myelination of neurons, it means that those neurons are getting insulated. So once again, we have the chemical signal coming in. If there's enough chemical, we generate an electrical signal that travels through this, like we could think of it like an electric cord, where it dictates the release of more chemical. So in a nutshell, that's what your neurons are doing. There are some neurons that are different. You can see this one. This is a picture right from your book. And you can see that on the end of this particular neuron, instead of this dendrite looking thing, this is where this is an example of a sensory neuron that can receive a different sort of information. And so this is a touch receptor, and it's sensitive to pressure, right? So stimulus from an environment, in this case, it's pressure, like on your skin. And when there's enough pressure, then that signal is communicated. And there are other neurons, like some of the neurons going through your limbs. Your neurons are throughout your nervous system. Some of them travel through your limbs like these really long, they have really long axons, you know, to get you to be able to move your body. But in a nutshell, that's how this is working. Any questions or anything that you would like to add if you've had this before that you learned elsewhere, anything?
If there's damage anywhere, are they able to heal?
Sometimes. So children have great capacity for healing. When people are older, sometimes, you know, people experience forgetfulness, memory loss, and that may come from that myelin that's damaged, but the body isn't as quick at repairing it in an older person. And there are some conditions. My nephew has a condition called Angelman syndrome, a very, very serious condition. And in that case, neurons in key regions aren't properly myelinated. There's not enough, and so signals don't get through. So he lost capacity to do even speaking. He can't really speak even. So this is an example of the electrochemical signaling. So we have two pictures here. First off, we're showing this is a picture of two neurons. This is neuron 1, this is neuron 2. So this is the neuron through which the signal is originally being generated. If the signal is generated and goes through this neuron to the axon terminal, then it is released into these little tiny gaps. You can see what the gap, it's not really easy to see it here, so that's why they made a bigger picture. But this neuron and this neuron aren't touching. Instead, there's a gap here between the axon terminals of one neuron and the dendrites of the next neuron, and that gap is called a synapse. So in this picture, this image is representing in large scale what is right in this little circle. And so this is showing the axon terminal. These little circles in here are called vesicles, and the brown dots inside those circles, this is neurotransmitter. So I'm going to just draw and write vesicles with neurotransmitter. And you'll recall that neurotransmitters, neuro, within the brain, transmitter, sender of signals. Neurotransmitters are simply chemical messengers. Let's see if I wrote this down. Okay, I didn't write that down, so I'm going to write it up here. Neurotransmitters are just the chemical messengers in your brain, like serotonin, dopamine. You've heard of these things. Chemical messengers in brain. So what's going on here is that this electrical signal has gone through the axon into the axon terminals, and this is the large picture. And because there was enough electrical signal, these gates opened and the neurotransmitter is released, and it lands in the synapse. This right here is the dendrite of what we call the postsynaptic neuron, meaning the neuron that's after the synapse. That's all that means. This is the presynaptic neuron. This is the postsynaptic neuron. And so the receptors in the postsynaptic neuron are receiving the chemical. So that's all that's going on there. And as long as enough chemical is released, the next neuron will fire and you'll be able to move your arm after lots and lots of neurons. So I'll just read what I wrote here. The messages received at the dendrite of this postsynaptic neuron via chemicals called neurotransmitters. If enough is received, an electrical signal called an action potential is created. So this is a term that you'll read about in your book. And I'm going to define it on the next slide. So the signal travels through the axon as a nerve impulse, jumping the nodes of Ranvier. So this is that node of Ranvier that I mentioned. So the signal is like zipping through here. It jumps, it zips through many times. Neurons are depicted with many of these little myelinated segments connected by the nodes of Ranvier. The neurotransmitter is released into the synapse from small vesicles in the axon terminals. And the big thing that I already highlighted once is that we're not hardwired. It's like there's no hard connections there. There's just this really beautifully dynamic system that's able to learn. Talk about the action potential, and then I'll ask if you have more questions. So this is such a weird word, isn't it? Action potentials and resting potentials. Because when we think of the word potential, someone has a lot of potential. It means like they have good gifts, and if they use them well, good things will happen, right? And this kind of means something similar. But this word potential is actually a word from physics, and it just means stored energy. Okay. And so the resting potential is just the energy that's stored in the membrane that's ready for action. So if we go back to, let's see, where should we go back to? If we go back to, remember I showed you right here is where the presynaptic neuron decides if there is enough signal. Well, the resting potential is they're always checking, is there enough signal this time? Is there enough signal this time? That's the resting potential. Like, there's potential that there could be something happening, but it's just a potential at this point. It's just resting, it's just waiting. So when there's not enough chemical, there's nothing going on, that neuron is just resting. The action potential is what happens when it actually fires. When the chemical messengers meet the threshold and there's enough push that all of that electrical signal is released, all that energy. So it's called an action potential because it potentially can cause an action, but it just means the neuron fired. And so because this science came from a variety of different scientific disciplines, these words are used in a slightly different way. So I'm not going to ask you about all the details of the polarization, okay? That's just the electrical language for what's happening in the chemical electrical interchange. I'm going to talk to you instead about what's actually happening. So let's look at this diagram. So first, right here, look at this gray line. This is the threshold. The threshold refers to the level of chemical signal that is needed for the neuron to fire. Or I'll say the amount, maybe the amount of chemical signal from the neurotransmitters needed to fire or release that electrical charge. Fire or create. We'll use that term, and action potential. So that's how much is needed. If the stimulus or the chemical signal, right, that's what we mean. It could be also the touch in a different sort of neuron, but we'll talk about the cerebral neurons now. Cortical neurons. If the stimulus is below the threshold, then there might be a hope of firing, but it will fail. There's not enough to completely cause a full firing, so the neuron won't fire. It's all or nothing. Either the neuron fires, releases its chemical, or it doesn't. And these are fails, so there's not enough chemical messenger, nothing happens. Depolarization is just the term for what happens when there's enough chemical. So now the chemical level reached, right here, reached threshold. So now the action potential happens. The whole electrical charge, the stored energy is released. There's a peak of energy release peaking out right up here. And then it's a big push, a signal, it releases, and then it goes back down. Right? That's the action potential. So the neuron's fired, it's done its job. It's released its chemical. And then after it releases its chemical, it goes into what's called a refractory period. So now the threshold, let me see if I can get a different color here. The threshold remains the same, you'll notice, right? The threshold is the threshold. But look how low the signal is. It's in the negative, right? It's in the reverse end. Look how much signal would be needed to reach this threshold now. During the refractory period, it is very unlikely that this neuron will fire, because it would take a lot of chemical messenger to reach threshold and fire again. So it needs to restore its energy. It's below, it doesn't have enough energy during this stage. Then its energy gets restored and it goes back into its resting state where it's ready for another action potential. This idea becomes important later when we talk about long-term potentiation. So now I'll ask what questions you might have.
So during the depolarization part, is that when as the chemical signal passes through the threshold, does the depolarization cause the action potential? Or?
Yes. If enough chemical comes in, and then the depolarization is what happens inside the electrical you know, the electrical energy is always like an imbalance of the electrons inside each molecule even. And when there's an imbalance, then the balance has to be restored. That's explaining it more than you need to know, because really all you need to know is that the chemicals come in, meets threshold, and if there's enough chemical, then the electrical signal is released, travels through the axon, causes the release of more chemical. You'll read more detail a little bit in your book. Any other questions? I was trying to think of a metaphor, but it's kind of a weak metaphor, but it's an all or nothing. Like either there's enough chemical for the neuron to fire or there's not. Like it's a firm line. It's like pass fail, right? So I was trying to think of it as like these little efforts, like he's got to generate a lot of energy to kick this ball up over the hill. And like he'll kick it, it'll go up, it'll roll back, kick it, roll up, go back. But then if he kicks it hard enough, it'll go up and it'll go all the way over. I was trying to think of it like that, but I think it's sort of a weak metaphor. But you can remember all or nothing. Like there's no half firing. Okay, so how do we know all this? A few different ways. We can use electrodes, tiny microscopic electrodes. They look like this, except this is a really big one. So they're really tiny, minute ones, because neurons are infinitesimally small. And so they're placed on the axon of a neuron, and the electrical charge then causes a certain pressure that the electrode can respond to, and it will go into something that's gauging the strength of the electrical current going through, and then going into like a computer or something to keep track of what's going on in the neuron at each time. So we can record it's called the magnitude of the pressure. So how great, how strong is the pressure? And then we can tell if each neuron is firing. So we can put little electrodes in. So this is some interesting work that I thought we'd discuss just a little bit. So we can have pressure on our skin. So right now, picture that neuron with that little ball on the end of it responding to your pressure. So what do you think happens when you push really, really hard, like when there's a lot of pressure? Do you think that the electrical charge is like stronger, or what do you think happens that indicates like to your brain, that's a lot of pressure? Do you think the action potential goes higher?
There's probably more chemical being released and building up.
All right. So inside the individual neuron, so would that be due to like, you know how there's that peak in the action potential? Would the action potential be going really high then to release more? What do you think?
I think that relapse part just fills up quicker and it shoots off.
It recovers.
faster.
The refractory period is shorter, maybe. Good hypothesis. Good. That's what originally was hypothesized by the researcher whose name I'm forgetting right now, did this work, and it actually was proven not to be true. So the action potential does not take on a different shape. Like the refractory period, there's still a refractory period that's about the same low level. There's still a peak that's about the same high level. So that doesn't change. But what does change is how rapidly it can fire. So in a way, you're kind of right. The refractory period is still just as low. Yeah, and so the whole neuron is just firing faster. So this little diagram is showing if there's light pressure, the neuron maybe is firing at this rate. This is the passage of time. Right. If there's moderate pressure, well, the neuron's firing a little faster. If there's a lot of pressure, it's firing really fast. So it's all happening a lot faster, but it's not changing the shape of the action potential. And so in general, you can think of this as all your neurons, not just your skin pressure neurons. The more rapidly the neuron is firing, the more you're experiencing whatever it is that that neuron is doing. So when you're thinking really hard, cogitating, working really hard in your brain, what's happening isn't any individual neuron changing its action potential, it's that the things are just happening faster. But the height, the level of chemical, none of that is changing, it's just happening faster. So your book posed the question in this way. It says, okay, we know magnitude of experience, we just talked about, you'll get more sensation if the neuron fires faster. We know it's related to the rate of firing. But what about the quality of the experience or the type of experience? In other words, how do we perceive light, sound, and so forth as different experiences? Like, if the neurons are all kind of the same and working in the same way, then how do we perceive these things as different experiences? And that's what they were. That's the way your book kind of offered this point, which was kind of interesting. And that brought to this really interesting principle of neural representation, which just means, I couldn't put the page because our digital book doesn't do page numbers very well. Everything a person experiences is based on representations in the person's nervous system. So if you're experiencing it, something has to be happening in your nervous system to allow you to experience it. There's no experience that doesn't come with something going on physically in your brain or nervous system. So we see patterns of neural activity in the brain that model some feature in the outside world. These representations are based on the connections, the quality of the connections, and the regions of the brain activated during a particular mental experience. And it includes what you're experiencing along with related ideas. And we're going to tease this last one out a little bit more. So you're experiencing sights and sounds. Like if you see someone walk by in the hallway, your visual cortex is active. But are any other parts of your brains active? Maybe your facial recognition system, maybe you have some judgments that arise based on something about that person. Or the speed they're walking, you probably assess the speed they're walking at. So there's a lot of different regions that are activated. So this is an example of that, but we are going to tease it out a little bit more. So this, your visual cortex is back here, right? So this is just your primary visual cortex. When information comes in through your eyes, it crosses at the chiasm, and then it goes back to this region. You also have a secondary visual system that comes into your emotional center, your lower levels. But in your brain, when you see something, not just that visual system is active, also areas related to, say, assessing the speed of the person, looking at what they're wearing, their judgments about linking it to other areas, and then linked to memories, emotions. So sensory information is not just processed as sensory information, it's processed along with a bunch of associated ideas. We look outside and we see it's raining. That piece of information is objective, but what other parts of your brain might be activated when you look outside and see it's raining? Do you feel any emotional activation?
Calmness.
Calm. Other people might experience more sadness. Does your mind go forward to things you would plan to do that you can't do? Or did you just begin to wonder if your windows on your car were closed? You see all the associated memories? Does your dog, get into his kennel, all those things? So that's the idea. But this stuff that we're coming to is really kind of interesting. So Hubel and Wiesel are pretty famous for work in this area. And this is I quoted their hypothesis. If action potentials and impulses are the same across neurons, that means that neurons must differ in terms of what they can respond to. So if they work the same way, then they must respond to different things. And so to test the hypothesis, they use these microelectrodes to see what neurons are firing when different stimuli are presented. And we'll look at how this was done on the next slide. And they found that some neurons are feature detectors. They fire only in response to a very specific visual stimuli. For example, there are some neurons that fire only when they see an oriented bar at the diagonal. There are some neurons that respond only when there's a bar or a shape or a line, something running in this direction, moving in this direction. There are neurons that respond only when there's a short something moving in this direction. And you can think that cats need to hunt, right? So they need to have a visual system that can detect movement. But now we discover that the neuron that detects something moving that way that's short is a different neuron than the one that detects something big moving that way. You know, different neurons. Tiny, tiny little cells. So let me see where we are here. Okay, we're going to move back to that cat. It's not on this slide. So what we call that is experience-dependent plasticity. And this is an interesting word. You can think of plastic as being malleable. You can shape it. You can heat it up and shape it into something else. If you've ever left something plastic in your car on a very hot day with the windows closed, maybe that plastic thing actually melted, right? So some basic rules: pathways that are used often fire often. If a pathway fires often, they're more likely to fire in the future. If there's a path in our brain that we're not using, your brain gets rid of it, so it can make use of those neurons and that energy for other purposes. And regions that we use often become larger in gray matter, and the general area where those neurons, gray matter refers to the neurons, the actual stuff of our brains, and white matter is the supportive tissue surrounding it. So gray matter is the neurons. So there's actually more, your neurons get bigger, more dendrites, more gray matter. This one right here, maybe you've observed this in yourself. I noticed this in myself. I played the saxophone in grade school and high school and even a little in college. And then, you know, maybe 20 years after that, I picked up the saxophone again. And this is what I found. My fingers still knew what to do. My mouth had no idea how to make that saxophone sound good. And so I could barely do what I used to be able to do. And that's because I hadn't used those pathways in so many years. I had not retained the knowledge. My brain said, you're not using it? Fine. We're going to put it over in this department. So we're going to talk about this more in the next lecture, but a little bit of teaser here. So this is just highlights some of what I said. So neuroplasticity is the brain's ability to change and adapt thanks to the fact that we're not hardwired. The structural plasticity is sort of like this. If you get a lot of gray matter in some part of your brain because you're using it a lot, maybe you need to use some thought processes for your job. You're using those all the time. There's more gray matter, which means the region of your brain devoted to that function changes. And then functional plasticity, brain functions move from a damaged area to an undamaged area. So if you damage, but it takes a lot of time. So if you know someone who's had a stroke and they've lost their ability to speak, as long as there's still some slight capacity left in their brain, they can, through hard work, retrain their brain so that their brain uses some of other neurons to take over the function of the damaged area of the brain. Talk more about neuroplasticity. Okay, a little bit more here, and then we can talk for another minute here. So experience-dependent plasticity is that idea that for our brains to be plastic and change, we have to demand things of it. And then we're going to get back to our vertical lines and horizontal lines in the kitties. So Hubel and Weasel again did studies where they actually took kittens and covered one eye for the first 8 weeks of life. And then they uncovered the eye. Any guesses or predictions of what visual capacity the kitten might have developed after the eye was uncovered?
The eye must have been sensitive to light.
The eye might have been, perhaps the eye is really sensitive to light. That's what would happen to us, right? It would be dark for so long. As adults, that would happen. Kaiba, I see, I don't know if you have any.
Yeah, I was gonna say, the eye that was covered would probably be weaker, and then the eye that was open maybe stronger, just
Ah,
because they were using it more.
Yeah, that eye might have been weakened through non-use. Why would that happen in terms of the brain, would you say, Kaiba?
I think I don't know if it has anything to do with it, but is it like the apoptosis, the neuron death or something like that? Of where if it's not being used, it eventually just it gets rid of it.
Yeah, well, yeah. And so what you're talking about in general terms is the eye that isn't used in adults, certainly. If we cover an eye for a while, the eye that we're actually using becomes stronger. Absolutely. And in us, we've already formed our visual pathways. So there would be a region of our brain developed to it. But thinking now about kittens, they're born with their eyes closed. And so no visual information is getting to their brain. Now, visual information gets to the brain, but only through one eye. Before any information gets to the brain, they can't interpret visual information. So when kittens first open their eyes, they really can't interpret any of the information they see around us. They have to get some experience. And so I don't know if I wrote it. So the answer to what happened is the kitten could not ever see out of the eye. Even though the eyeball itself, the retina, the nerve at the back of the eye, the pupil, all of that was undamaged. There was no damage to the eye, but the brain never developed the pathways it needed to be able to use the eye. And in kittens, if that kitten doesn't have any experience with the visual system for the first 8 weeks of life, they can never develop it. So once again, the eye is just like the import, right? I mean, that's just like the port where the information goes in, but the brain has to make sense of it. And without an accompanying brain region, this kitten can never make sense of visual information from that eye. This kitten lost the opportunity. And that's called a critical period. A period of time during which an individual must have some experience in order for a neural pathway to form. The kitten never had visual experience. The neural pathway did not form. So what is this critical period? For vision, the critical period is 3 to 8 weeks. After that, no. In humans, like some premature infants born with cataracts or like a clouded lens in their eye, we have 6 years for at least some vision to be possible. But if no vision in the first 6 years, there can be no information from that eye. And that's why they right away operate on an infant that has cataracts, because otherwise the brain to interpret it won't develop. That's why even in this infant, we put corrective glasses on so that the brain linked to eyesight can develop properly interpreting visual information in the world around. And when they do surgery on an infant, if they do it one eye at a time, then after they correct the bad eye, like they correct a bad eye and open it, and then after a while they correct the other eye, then they cover up the good eye so that the infant has to rely on the bad eye Because if they didn't, the infant would just continue to go through life and rely on the good eye, because that pathways became very strong. You have to then weaken those pathways through non-use to get the other pathways to develop. It's pretty wild, isn't it, these critical periods? So this brings us back to these feature detecting neurons. Are feature detecting neurons dependent on experience? And the answer is absolutely. And the experiment, again, that Blake Smith and Cooper did is they reared kittens in a space with only vertical black and white stripes on the walls. That's it. There were no horizontal lines, and you can see that they put a cone on the kitten such that it couldn't even see the floor and the ceiling line. So all it could see were vertical stripes. Now we know there are feature-dependent neurons. So how did they test after a while whether the kitten could detect different kinds of lines? Well, they did it through what the kittens would respond to. Kittens raised in this environment would bat at a vertical moving stick, but couldn't perceive horizontal objects. These animal experiments, if you're like many of us are bothered by this sort of thing, and you should know that we do have animal rights protections in place in this time, so that anything that's done with animals, a person has to demonstrate the benefits of the work that outweigh the harm to the animal. And so why? The neurons that would normally respond to horizontal lines were usurped by the vertical feature detectors because there were no horizontal lines. So the vertical line area said, hey, all right, we're going to use you, you know. If you think about when someone goes blind in adulthood, then all of a sudden the brain, their huge visual cortex, and the whole regions of our brain devoted to processing vision, all of a sudden those neurons are going to waste. There's no more visual information. What do you think happens in the brain? I mean,
You start pruning away, like you said, or start going, or being used for other types of
being used for other types of
functions.
sensory information often. I mean, blind kinesthetic information. Like, have you ever wondered how Helen Keller could detect someone writing letters in her hand and figure out what her world is through this? I mean, it's because she had no sight or hearing. She had very strong sensory system, right? Very strong olfactory system. Probably taste too. Yeah, so it's kind of good that our brain can. Do you know that really famous scientist, I think Stephen Hawking, I always get Richard Dawkins and Stephen Hawking messed up, but I think Hawking, he's a very famous physicist. But as a young man, when he was just studying the sciences, he developed a neurodegenerative disease that caused him to lose all function to move his body. So he lost his body, he just had his brain. I think his cognitive regions took over some of those regions that used to be devoted to moving his body, and that's why he can come up with some of this brilliant conclusions.
Did it also include speaking as well?
He couldn't speak very well, as I recall, but his colleagues could understand him, but somehow he could still write, I think, but typing maybe?
Yeah.
Because his thinking wasn't damaged. What time are we getting to here? 2:30? We go till 3:40, and I have this really cool content on. All right, let's go for another, say, 10 minutes and then take a short break. We actually have feature detecting neurons just for faces, because in our social world, it's so important to recognize faces. And I wrote this, Oliver Sacks wrote a book called this. So some neurons are assigned specifically to face recognition, and if someone has damage in this reason, they cannot distinguish faces from inanimate objects. I posted this video for you, but we're gonna watch a very short segment of it. Someone with a condition called propagnesia, and he can't see faces, and this is what his experience is. I need to just activate the sound in our Zoom meeting. One moment. So,
Do you know who this is?
just a minute. Did you guys know who that was?
Oprah.
That was Oprah. Okay.
She's pretty, a very symmetrical face, nice teeth. I don't know who she is though.
Oprah?
Oh, darn it. I know who Oprah is. Oprah's got such a distinctive voice. Who needs to recognize her face? But yeah, sorry about that, Oprah.
Paul knows who Oprah is, but when he sees just her face, he draws a total blank. That's because his face network, the areas of his brain responsible for facial recognition, developed atypically. It's a condition called prosopagnosia, or face blindness. It's more common than you'd think, affecting as many as 1 in 50 people. But its prevalence has only been understood in the past 20 years. And the fact that it is so little known often leads to painful misunderstandings.
Some people in my life just developed a serious dislike of me. Oh, this guy's such a jerk. We've talked several times and he never acknowledges me.
What people on the face blindness spectrum need is for you and I to understand what they're experiencing. So let's do an experiment. For those of us without face blindness, perhaps the best way to understand how Paul looks at a face is to look at these faces upside down. Even though you can see the individual facial features, you might not immediately recognize who these people are. At least, not until we turn them right side up.
Do you recognize some of these?
If you can recognize a face, then it's a bit like you can fly through life on autopilot. But what if you couldn't? Imagine the office holiday party. You're surrounded by people who you see every day, but you can't tell who is who. Are these people smiling because they recognize you, or just being nice?
There's a certain amount of fear that somebody's going to come back and say, what kind of idiot are you?
Even in a crowded room, you're left in a world of one. This fear also occurs in the most private settings. Take what happened just a few years ago. Paul was visiting his favorite cousin, Rose.
I knew she was going to be there. I knew she was pregnant.
But when he walked through the door,
About 15 feet away, there's this pregnant woman. I just jumped and said, that's probably a friend of Rose's who also happens to be pregnant. They have a lot in common. I offered her my hand. I said, hi, I'm Rose's cousin. And she laughed. And as soon as she laughed, I knew, oh, okay, that's Rose.
So this is a very good video, but for the purposes of our class, they don't really go into what's happening in the brain, so we're going to talk about that a little bit more, not just in the next slide, but over the series of the next content. All right, so in any case, this is just evidence that there's this particular neuron that responds to, this is with the primate, that responds to face stimuli, it fires. And it doesn't fire at all. In fact, it's a little below on non-faces. So feature detectors for faces. And Oliver Sacks describes a case of prosopagnosia. I don't know why I struggle with that. In a book of the same title called The Man Who Mistook His Wife for a Hat. And this damage happened later in life, so it was disturbing for him, obviously. Well, that's not supposed to come yet. Okay, this idea is in your book. You can read about it more. It's called Hierarchical Coding. And it means that when information comes into the brain, I'm going to just slow down, I don't have it, that they come from more general to more specific neurons. So for example, that cat whose neuron fired at a vertical, shape moving in this direction or something like that, it would start out at those neurons. But then it moves in and makes more sense of those neurons, so into a more complex neuron, until finally these things are put together to form the complete image of the mouse or whatever it is, or the face or whatever it is. So that's hierarchical because it starts out as just those basic feature detectors for very simple aspects of the feature. All right. And the sensory code is the way neurons represent characteristics of the environment. That's just defining a term. And specificity coding. The idea that an object, any object we see, chair, table, anything, could be represented by the firing of a specialized neuron that responds only to that object. So this is a theory, okay? Do we have specificity coding? You can already see because of the X that came up too soon that we don't. But this idea, if it was true, if you can see the image behind the X, you'll see that we're looking at 10 different neurons. And only number 4 fires when it sees Bill. Here, we're looking at the same spectrum of 10 neurons, and only neuron 9 fires when it sees Mary. We're looking at the same 10 neurons, and only number 6 fires when it sees Raphael. That's what specificity coding is. And it's not likely the answer to how these things work, but this is one theory about it. Do we have a neuron for every face we see? Do I have a particular neuron that fires when I see Virgie, along with any other associated regions? And the answer is no, and you can see I didn't phase this in properly either. We have 2 other possibilities, this population coding, which means that maybe there's a lot of different neurons and that form a certain pattern when they see an individual. So like maybe a whole bunch of neurons show this pattern when they see Bill, and there's this pattern when we see Mary, and this pattern when we see Raphael, and you know, this neuron number 2 is involved a little bit in both Raphael and Bill. But the answer to that is sometimes when it's necessary, but usually we don't need that many neurons for something that simple, and we usually rely on sparse coding. So that means that as few neurons as possible are engaged. And so it's not just one neuron, but it's as few neurons as possible. And that seems to be the dominant model that's supported for things like recognizing a person. And so that's why I circled it. All right, and let's see here. I'm going to decide where we should stop and how we should integrate our next content. So, and this is, let's go a little bit further. We'll go until you have to leave, okay, Kyle? So we'll both make note of our time. So when we talk about perception and memory, there's a question that we might have. Let me see if I wrote it here. Yes. Okay, so perception, we see something that's happening. We're perceiving it right now. And memory, well, we can each remember what was happening a few minutes ago or a couple hours ago or what we had for lunch, right? So both of these things are stored, they're kind of image representations, right? Things that are happening, stored very often as images. Here we have the perception actually happening, and here time has passed and we have the memory. And so the question that was asked is, how can the brain distinguish perception from memory if they're both images of the same thing? And so if we activate in our minds, think back right now to talking to someone this morning before you came to campus. Now there's a memory, and you got a picture in your mind. How does your mind know if you got the same picture? How does your mind know that that's a memory? And not happening right now, because the picture is the same, right? How can your brain tell what's happening right now from a memory?
From the region it's coming from and the process your brain goes through to bring it back.
Okay, possibly, but let me make one more point. You know, the representation of the image is the same neurons. The features of the image is that same specific image, like the same eventual firing that was happening when you first had the perception.
And I think it's the source, because if it's happening now, you're getting it from this section, but if it's a memory, it's a different part of your brain that's pulling it up.
You're right in a way. It is sort of like the source. So it's the same region of your brain when it actually happened, like the picture of what, this is the example I'm giving. You saw this burglar, okay? And here's your eye, you took in the information, this is actually happening, you saw this burglar, all right? Now think of hierarchical coding. The first neurons to fire are like the ones who recognize, we haven't talked about it yet, who recognize arms and legs, the ones who recognize vertical lines, horizontal lines, movement, really simple neurons. Then it goes into more complex neurons and then more complex neurons until finally, so this is what I was trying to demonstrate that with. So here's the simple neuron, the vertical line, horizontal line. Here's a simple neuron, a smell. Simple neuron, a vertical line. Simple neuron, motion. But then they all combine to make the complex neuron that is the image of this burglar. So this is the same image when you first saw him as when you do now. But during the memory recall, only this last image is brought forward. All of these preparatory neurons out there. And so that's one way that your brain can tell. And you also are probably conscious, I tried to make him a little blurry, that your original perception was probably more clear than your memory. It's a little bit blurry when it's just in memory. So it's an interesting question, isn't it? You know, never thought about that one before too much. This is a good stopping point. We're going to talk about localized representation. So let's take a break. I'll set my timer for, well, we usually take 10 minutes. I think we can use it. Set timer for 10 minutes. And think of any questions you have. We'll start with your questions when we get back.
Every time I move this, when this little side table moves, it kind of freaks me out.
I know, I don't know how to, last spring, I was successful in getting them to bring chairs into this room. Now, someone took the chairs out. So I actually nabbed this one out of another room and so far no one has said anything to me, but it's crazy how like, really it's stingy with chairs here.
Alrighty, thank you.
Thanks for being here while you could, Kyle. And tell Dr. Denny that, you know, we're fighting over you.
I know, I wish I could just go over right here and sit there, but.
I have one ear. You could do your dichotic listening test and listen to this class in one and.
But my boys come back at 4:40, so
Yeah.
they would be home alone for an hour.
Oh, no. So is this stuff that we've been doing kind of new to you, or have you had this kind of content a little bit before? We're having a break.
Yeah. Yeah.
Okay. All right, let's get back to the video. Hi, everyone. Hi, you guys. If you're back online, pop on in here. Let me know you're back. Wow, that's a great view of.
Antelope Canyon.
Is that Antelope Canyon? Oh. Is that down in Canyon?
Page.
Oh, that's okay.
It might be on Page, yeah. I used to be a tour guide there. It looks familiar.
Oh, really?
Yeah.
It looks like a place I was at called Buckskin Gulch up by.
Oh, that's a good one too.
Yeah, I backpacked in there. Hi, Abby. Hi, Ashley. Hi, Rainey. Okay, I think we're getting close enough to get ourselves started again. So what questions or comments or anything do you have, just so these classes where I'm doing all the talking aren't my favorite kind, so I'd love to hear anything from any of you before we get back into some new content. Do you like this stuff? Neurocognition? Anyone?
I would say It's interesting, but also really complicated.
Yeah,
And to answer your question when you first asked, this is my first time taking something like this. So it's all kind of new.
it's really new for you.
Yeah, it's really new. At least a bit of it might be from Psych with Jeremiah Barber,
Okay,
and a little bit from Bio,
yeah,
160. A little bit of that, but not more delved into it.
a little bit more, yeah.
So this is like.
That's good though. That's the way it should be. You should be getting little pieces and building your knowledge that way. That's a good way to build. And this is, yeah, that sounds okay. Thanks for chiming in there. I appreciate that. All right, we're sharing my screen, so let's get back to, I'm hoping to cover this localization a little more quickly so I can get into this fun stuff. So all we mean by localized representation is this question, and we'll answer the extent of particular regions of the brain areas, physical areas devoted to particular functions, okay? And there's a little video that we could watch about Phineas Gage, and I actually thought that I would show it, but our time doesn't allow it right now. So let me just pull up a little picture of what happened to Phineas Gage. If you're not familiar, you probably learned this in Intro to Psych. We'll just do this basic psych. So he was Phineas Gage lived back in the 1800s. And he was the foreman actually, because he was really responsible, a really good guy. And he was the foreman of this construction crew. I think they were building a railroad. And something went wrong and there was an explosion. And this big post that he's holding went right through his head, through his brain. You can see it came, let's see if we can get the angle it came in. Right here, you can see how it went into his skull that way. And he didn't lose consciousness. He didn't pass out. He pulled the spike out and went to the hospital and continued his life. But what went wrong is that certain things changed. He lost his sense of responsibility, for one thing. So he could no longer like go to work on time or skip out of work. He started drinking. He lost the ability to control his temper, so he started to be really like angry and a little bit volatile. He became very indecisive. He couldn't reason things out to come to a good decision. And in the end, he could no longer work as a foreman on that crew. He worked still, but a different job, and he was a different man, according to his friends. So what do we learn from this sad case? So if we look at the region of his brain that was damaged by this pipe, you can see that it went through his frontal lobe. You can see that he lost his eyesight in that eye. That makes sense. We can understand that. But that helps us to understand what the frontal lobe does. And the frontal lobe, especially the prefrontal cortex, is like our executive function. It allows us to regulate emotion. It allows us to make decisions. It allows us to balance things out. It allows us to prioritize. And he lost all of that, or a lot of it, in that accident that happened. And so one way, even though it's really sad, but one way that we can learn about what happens in different regions of the brain is by looking at people who through illness or birth anomalies or injuries lose some part of their brain. So that's one way we learn. We're going to learn about 3 ways that we understand. So localized representation, again, just means that specific functions are served by specific areas of the brain. So our cerebral cortex, what's meant by that is that very thin outer layer where all the wrinkles are in our brain, okay? That's the cerebral cortex. The subcortical areas are all the other areas beneath that outer region where all the wrinkles are, where the gyros are. That's related to a number of different functions, including emotions. And this field where we study people who have had injuries is neuropsychology. And you know that there's a neurologist who studies that for the purpose of healing, and neuropsychologists study it for the purpose of learning. So here's some evidence from neuropsychology. An area called Broca's area. You can see it here, and it's usually taught in conjunction with Wernicke's area, and we'll talk about both. So Broca's area is a region of the brain that when it's damaged, when someone has a stroke very often and gets damaged to this particular region of the brain, their speech, even though they know what they want to say, this is really frustrating for the person who's suffering because they know what they want to say, but they can't do it. They can't find the words, they're slow, their speaking is labored, it's not grammatical anymore. They can still think okay though, so it's gotta be very frustrating. And this is an example of someone who had a stroke while in a hot tub, who was asked about his stroke, and his words were, all right, stroke, and Tuwana guy, hot tub, and 2 days when hospital. You can see how this man knows what he needs to say and can't do it. And so this area is related to speak production of language. And by contrast, Wernicke's area is a different brain that's active when comprehending language. Any deficiency, inability to speak or communicate with language is called aphasia. And so Wernicke's aphasia, we had Broca's aphasia, now we have Wernicke's aphasia. They still have their grammar, and they can speak fluently, but it doesn't make any sense. And so this person who had damage after a stroke says, it just suddenly had a pfeffer, and all the pfeffer had gone with it. It even stepped my horn. They took them from earth, you know. They make my favorite 9 to severed, and now I've been habbed all by the fortment of my annulment, which is now forever. So the comprehension, the ideas underlying the expression of language don't seem to be there. The comprehension of what ideas are represented by language. And they're both linked to damage in particular areas. So this is a pretty sound conclusion that these regions of the brain serve those functions. Now, we do know that it's not just these regions of the brain, that along with these are other regions that help us with language, and quite a few regions, in fact. But more evidence from neuropsychology. I'll just kind of highlight these. You can read about them more in your book. Actually, the next slide is what I'm going to highlight. We know our occipital lobe is where our primary visual cortex is. And we know that if soldiers in World War II had damage in this region, they would either lose their sight, depending on the damage, or lose their sight in a corresponding region, a region corresponding to the area of the damage. So if the damage was on the left side of their occipital lobe, they would lose the ability to interpret visual information from the left side of from the left eye, or in the left field. Our neighbor. Temporal lobe, you can see on the side here, linked to hearing, our auditory cortex. The parietal lobe, we'll look at this a little more closely. It's linked to touch, pressure, pain, any senses that come in through our body system, our system of touch. And the frontal lobe, as we've already talked about, is this coordination of senses and higher cognitive functions like thinking, reasoning, prioritizing, deciding, regulating emotion. So how do we know all this stuff? Have any of you ever had an injury to your body, like your knee or something, like, and you went, when you hear about an athlete who gets an injury to their knee or something, they go in and get an MRI, not an fMRI. And the MRI, like it uses magnets, and it actually is able to take pictures of very thin slices of what's inside there. And when I say thin slices, I don't mean they slice the knee, but I mean it can get very thin layers, pictures of very thin layers in this three-dimensional object called the knee. Well, we can also do MRI on the brain. And so we can just do a basic MRI and see the structure of the brain. But if we do an fMRI, F referring to functional, with that lowercase f in the front of it, then we can see where the action is in the brain while someone's doing something. So the person still has to lie. They go into this big magnet. So they lie on this bench and they have to be very still. Their head is held, in place. And then they're pushed into this big magnet. It's very loud, I understand. I've never been in one. And I'm not going to test you on this, but how it works is when there's an area that your neurons are really active, it's firing a lot, it requires more oxygen. And you know that the hemoglobin is in the blood is what carries oxygen. And when these two are combined, it's more magnetic. And so this oxygenated hemoglobin responds more strongly to the magnetic field of the MRI. So when we get these pictures, we can see where the action is. And usually, last class, I think I made a little joke saying now everything is about the little blobs. And that's how some people who aren't in neuropsychology feel about the fact that it seems like many things that you want to demonstrate in psychology, now you want to have an MRI to support your hypothesis. And usually yellow is the most active region of the brain. And so since this person's in an MRI, we can put a little screen in front of them, or we can put something on their eyes and put images in their eyes or ask them to do certain things like push a button, certain things that don't move their head. And so then we can ask them to do something and see what part of the brain is involved. So that's how we know. So through that, we learned that we have this whole region of the brain in the temporal lobe that's called the fusiform face area. And that's the same area where that one neuron is that recognizes faces. Remember the face-activated neurons? And what I meant by that is there's not one neuron that recognizes faces. I meant they were recording single neurons to discover the neurons that were feature detectors for faces. And it's the same area where if someone has damage, they get prosopagnosia. So the fusiform face area, I know that's on one of the quiz questions. This is really weird. We have a region called the parahippocampal place area, PPA. And that same area is activated when a person is looking at indoor scenes like this room and outdoor scenes, and even looking at this room if there's no furniture in it. And it's believed then that it's linked to spatial layout. So people, meaning the way you navigate space. And people who have damage in this area would have trouble navigating space in a real world, three-dimensional world. We have another part of the brain that's activated specifically for bodies and body parts, but not faces. The extrastriate body area, EBA. And this is a comment that back during COVID, I heard from a couple different people that when you're just meeting with people on Zoom, and then you meet them in person, you might not recognize them. And why is that? Here we see our faces, even assuming they're not wearing a mask, we should be able to see them through their faces, But sometimes we can't. And it might be because this other region, because we also rely on the way they move their body, the shape of their bodies, and other features that we can't detect by just seeing someone's face. I find that all the time. I don't recognize students that I've only met on Zoom. It's unfortunate. And then there was concern when all this was happening about babies born during that time, and how most of the people that they were meeting had only partial faces, only eyes. And how would that impact brain development for facial recognition among those infants? Interesting question, right? So we all know a lot of babies and we think they recognize us, so I think we're okay. We may have that 6-year time frame for developing those kinds of features. They also gave people movies in the fMRI. And if you were a grad student in this lab, this would have been your job. So they had to code every single moment of the movie. And so like if you were a grad student, you'd look at this particular scene and you'd say, okay, what's in here? There's desert, there's sky, there's cloud, there's brush. What's in here? Woman, talk, gesticulate, book. You'd have to code every second of that movie and you'd all have to agree on it too. And then they would code they would look at what regions were active at every stage. And what you notice is that there's active regions throughout the brain for many, many different things. It's not just one region. And that kind of throws this localization idea kind of off the rocks here a little bit. And so we resolve that by understanding that every experience is multidimensional. We talked about this earlier. If you see a face, there is the initial processing in the visual cortex in the occipital lobe back here. There is some activity going on in your FFA, your fusiform. But there's also an emotional reaction, maybe. There's also maybe an evaluation of their attractiveness. There's also maybe an awareness of where they're looking, right? And other things, who they're with, how they're moving. And so we consider that all of our experiences aren't just one-dimensional, right? We're not just a camera recording images, but we're pulling up a lot of other experiences. And so because of that, many brain regions have to be activated, and that's called distributed representation. So each experience is represented in distributed areas throughout the brain. Now, I think that for the sake of the other content I want to share with you, I will post these slides, but I'm not going to continue the lecture. I'll talk to you about how an example of this is memory, how another example is language, and you'll read this in your book, and then there's a summary slide that I'm not going to go through with you. So that's what I have for you in your slides on this chapter, and your quiz is only based on the chapter, okay? And what I shared with you in these slides, because most of what I shared with you is from the chapter. I shared a little bit more, but it's not on the quiz